see that it is 3:30, and so folks will continue to join us, but if you are ready, Amelia and Alex, we'll go ahead and get started. Okay.
Yeah.
For everybody online, thank you so much for joining us. My name is Cinnamon Moffett, and I'm the Research Program Manager at Oregon State University's Hatfield Marine Science Center, and I will be your host for today. A couple of things about Zoom, which you all know, but we do have your cameras, mics, and screen shares turned off. So please put any questions you might have into the chat box, and we'll work through those at the end of today's presentation. If you need closed captioning, that is now activated, so you can click the CC at the bottom of your screen and you will get closed captioning. Also wanted to let folks know we're recording this and it'll go on the HMSC website under the past seminars page, probably in 2 days or so. Give me a few days to get that processed and get it posted. But if anybody is unable to join us for whatever reason, blizzards or any of those other things, please feel free to share the link in a few days. Couple of really quick announcements. Today is a busy day because tonight we also have a Science on Tap. So if you are interested in joining us, we are going to do a virtual Science on Tap tonight at 6 p.m. where Mauricio Cantor with the Marine Mammal Institute here at Hatfield is going to be talking about how innovative feeding strategies shape dolphin society. So that will be a really interesting talk. Another announcement is we are doing a green team, HMSC green team beach cleanup this weekend on Saturday. So if you're interested in that, go ahead and go to the HMSC website and go to the green teams page and you'll get information there. And if I get a chance, I'll put the link into the chat. The other thing I wanted to promote is next week's seminar on April 28th. Matthew Johnston, an assistant professor with the electrical and computer engineering at Oregon State University, is going to be giving us a talk about sensors in the wild, packaging, power autonomy, and other challenges. So that will be a really interesting talk, and we're excited to kind of have some engineering folks start to join us at Hatfield. That particular event will be on site in a hybrid format, so both online and in the room at the Marine Studies Building Auditorium. So feel free to join us for that. Yeah, and if you need any other information about upcoming events, you can go to the HMSC homepage and scroll to the bottom and you'll get all the information about things that are happening and how you can log on. But we're all here today for today's speaker. And so I'm going to actually hand this off to Alex. Alex is a postdoc in the Big Fish Lab. You all have met her recently when she did her own seminar for us. And she is the one that invited Amelia, our speaker today. So Alex, I'm going to hand it off to you.
Awesome. Thank you, Cinnamon. I'm really excited to introduce Dr. Amelia Munson today. She's a friend and colleague of mine, and we have a lot of similar research interests as well. So just to give you a little bit of information about her, she did her undergrad at Reed College in Portland, Oregon. And while there, she worked with Dr. Susie Wren on mouth brooding cichlids, which is where she first fell in love with fish. She then got her associate's degree in zookeeping and worked in a couple of different zoos. And she accumulated a lot of really good animal stories, which I can confirm while there, but ultimately missed research too much. And she got her PhD at UC Davis under Dr. Andy C., which is where I was fortunate enough to have met her. And then she moved to Scotland very recently to do a postdoc in Dr. Shaun Killen's lab, studying how elevated temperature influences physiology and social behavior in fish. And with that, I will let her take it away.
Thanks, Alex. It was so fun hearing about all the stuff that you guys are doing in Oregon. It seems like you've got a lot of events going on, which is awesome. So as Alex mentioned, I did my PhD at Davis, and a lot of the stuff that I'm talking about here was part of that dissertation work. So I'm currently in Glasgow, but definitely a lot of this definitely reflects the guidance of Andy C. and my PhD. So we're going to be talking about a variety of different topics that generally explain or attempt to explain how experience shapes variation and consistency in fish behavior. So when I started my dissertation, the sort of first question I had was this really general question about just how do animals respond to things? And I think a lot of people who study animals, that's sort of what their research can be boiled down to. And sometimes we know we can answer this pretty well. If you're imagining we have a gull and a little fish comes around, we might say, okay, the gull might eat that fish. And we can guess that based off of previous interactions we've had with gull, previous research. We have a pretty good idea of what they're going to do. However, increasingly, there are novel objects in an animal's environment, things like trash, and we don't necessarily know how an animal is going to react to it. We can sometimes guess based off of what they do in other circumstances, but using an animal's experiences may help us inform how they're going to respond to novel items, which are increasingly becoming a problem. This was a question that I was really interested in, but I was less interested in these sort of recent experiences. So less thinking about like, okay, he just ate a fish, that fish kind of looked like the garbage, maybe he'll go after that gull. And sort of more interested in these general patterns of behaviors that animals have. So this is me recently hanging out with my dog, and you might be able to imagine, oh, look, based off of recent experience, it looks like she really likes animals. Maybe she's going to do something with animals. But I really cared less, but again, this recent experience, and really wanted to sort of dig down and understand what is a creature at their core. And a lot of times these sort of core essences of animals come from experiences that they have during their development. So what they're experiencing when they're young and how that shapes them when they're older, and how that sort of leads to the way that they approach the world more generally. So spoiler alert, I was also super into animals and exposed to a ton of them as a child. And when I was thinking about this idea of how do you understand the general way that an animal approaches the world to better predict how they're going to respond to novel objects in their environment, I got really interested in thinking about animal personality. And some of that comes from my advisor. He is a big proponent of studying this and thinking about it as a sort of framework for understanding animal behavior. And if you're not familiar with animal personality, I think it's really helpful to sort of go over some terms because it is a common issue in the field, as in many fields, that words mean slightly different things and they mean different things to different people. So I think it's good that we're all on the same page when I'm talking about these issues, because it's going to come up a bunch in my presentation. So when I'm talking about personality, what I'm talking about are behavioral types, which are just the idea that behavior of an individual is consistent across time. So for example, if you have two little fish, you might have an individual that is consistently sort of more bold and an individual that is consistently less bold. And if they're bold at time point A, you can predict that they're also going to be more bold at time point B than a fellow fish. We also talk about behavioral syndromes, which are relationships between behavioral types. So you might have an individual that is more bold and also more aggressive, and an individual that is less bold and also correspondingly less aggressive. And these traits tend to be related in a variety of different species. And importantly, consistency has a bunch of benefits. So there's research to suggest that in some species of fish and other animals, females will actually make mate choice decisions not based on behavior per se, but on consistency level. So they just prefer males who perform more consistently, potentially because it can tell them something about how they're going to behave as a mate. Consistency also has benefits in terms of anti-predator avoidance, as well as when you're coordinating behaviors in groups. So there's lots of sort of benefits for an individual to behave consistently across time. When we're talking about animal personality, it's, I think, really easy to sort of tell a story and anthropomorphize an animal, and it gets really easy to like see ourselves in animals and say fun things. But really what we're talking about is a mathematical thing. What we are talking about is behavioral repeatability. And I want to spend a little bit of time talking about this because, again, it's going to become really important when we get into my research. Behavioral repeatability is a little complicated because it sounds pretty straightforward. And I think a lot of people feel like they have an intuitive grasp on it, but are forgetting what the actual measure is. Because repeatability is actually the measure of two different measures of variation. So take, for example, if I have a bunch of fish and I am measuring some aspect of their behavior, I might be measuring how fast they swim, how much they swim after they see a predator, any of those things. You're going to have variation. You're going to have variation among the individuals. So some individuals are going to swim more or swim less, swim faster or swim slower. And if you measure these same individuals at another time point, you are also going to have within individual variation. My mouse not working. You're going to have within individual variation. So an individual may have more or less variation relative to the other ones. Some of them may behave really similarly to how they behaved in the past, and other ones may behave totally differently. So you have these two sort of measures of variation. And repeatability is just the among individual variation divided by the within individual variation plus the among individual variation. So your total sources of variation and how much that is due to the among individual variation. And again, I think this is something that like gets kind of complicated because I put a fraction on the thing, but there's words and a lot going on. So it tends to get complicated. But the important thing is that when we're talking about repeatability, it's not just a measure of what one individual is doing. It's a measure of sort of how they're relating to their group. In order to have high repeatability, you would need to have both high among individual variation relative to lower within individual variation, a bigger fraction. And again, this can get a little complicated and people, I think, sometimes kind of zone out when I start talking about it, but it's actually really the basis of like every TV show that we watch that has multiple characters. And I feel like this is one of those slides where like whichever like TV shows I picked is probably going to age me, because I bet you could get a really good idea of how old I am based off of like the time span of TV shows that I think are fantastic. But related to repeatability, all of these shows that have lots of individuals in them, they are relying on high among individual variation and low within individual variation. So if, for example, we take Parks and Rec, we know that there's high among individual variation. Leslie always behaves differently from Ron, and there's low within individual variation. Leslie tends to behave the same episode after episodes. That's why we like it. We can predict what she's going to do, and we can predict that it's going to be different than Ron. So even things like breakfast choice become a measure of personality in these, right? Ron is always going to eat lots and lots of bacon, and Leslie is always going to eat lots and lots of waffles. And so that they are consistent in themselves, but they're very different from each other. Versus something like hating Larry is not a personality trait because all of the characters don't like him. So there's really low among individual variation. So even though they're consistent internally, because it's not different, we don't really consider that a personality trait. So again, this idea that the characters that we are looking for are different from each other, but consistent within themselves, that's what makes a really strong personality trait that's easy to follow. And that's the same type of thing we're looking for when we're studying animal personality. But why are we talking about repeatability, right? This was supposed to be a presentation where we were talking about development and how experiences in development affect the way that an animal responds to something new in its environment, right? Let's get there. So let's say, for example, if we have a bird, and ultimately I'm interested in understanding how its experiences shape how it responds to change and using personality as a way to understand those things. And oftentimes when I say that, what people think I'm going to explain to them is what is the perfect personality trait for an animal to have to be better at responding to environmental change. And unfortunately, I am not going to answer that. And I honestly think it would be a really hard question to answer, because if we imagine this little cedar waxwing, who is a very bold individual, that may be good if, for example, the environmental change is a new source of food. If he's super bold, he may be more likely to eat that food source first, be able to take advantage of it, and get all of the associated fitness benefits. But if the environmental change is something like a new predator in the environment, then this little waxwing who's still bold may be less scared of this predator and more likely to be eaten first. So there really isn't one perfect personality trait to protect an animal against all forms of environmental change. And I think that's one of the reasons that environmental change can be so challenging, is that different changes select for different individuals. So when I was thinking about this, the thing I got really interested in thinking about was whether or not it was still beneficial to be consistent. Traditionally, if an animal is consistent, that may be because the environment it is in was relatively consistent. But if the environment changes, it may no longer pay to be consistent, and it may instead be beneficial to be more flexible. And I'm going to argue a little bit later in my presentation that consistency is key to understanding responses to environmental change, but it's a complicated story as to when it pays to be consistent and when it doesn't. So just keep all of these things in your mind as we're moving forward. One of the things that I think we need to chat on just briefly is what determines your personality. If we're talking all about repeatability and the way that development affects us, does development even matter for personality? And it probably does. This is that classic nature nurture slide, and the answer is always that both of these things are pretty important. There is evidence to suggest that if your parents are one type of personality, and that can be both their level of repeatability and their sort of what their average behavior is, you are more likely to be that too. And this was based on a large meta-analysis over multiple studies, that there definitely does seem to be some heritable component to personality. And that is particularly actually prevalent when you look at personality, that the sort of repeatability and consistency measure is actually more repeatable than one individual behavior per se. But experience also matters, and experience during particular times in the life may matter particularly strongly. So there's evidence that temperature, immune challenge, nutrition, and predator exposure have all been shown to not just affect how an animal behaves, but how repeatable they are. So for example, I conducted some work with sticklebacks with a postdoc in my lab, Marcus Michelangeli, where we measured the social behavior of sticklebacks twice, and then housed them either in groups or alone, and then remeasured their social behavior twice. And we found that individuals that were housed alone were more social after we tested them a second time, but the repeatability had changed drastically. Individuals became more similar to the most repeatable individual in their tank. That not only was repeatability important for how others behaved, but it also affected the overall repeatability of each individual within the tank. So it seems like things like even who you're around and what your social conditions are can affect things like repeatability. One of the factors that seems to be really important is predation threat. So there's evidence across taxas in fish, mammals, birds, and inverts that predation, again, doesn't just affect behavior, but it affects how repeatable individuals are. So some research that I conducted with a variety of students in my lab on mosquito fish. We tested 4 different behaviors of mosquito fish in a variety of turbidity levels. So sort of how much dissolved matter is in the water. And we also tested their response to predators. And what we found is that mosquito fish were the most responsive to predators at intermediate levels of turbidity. And this actually is what we expected. So when the water is super clear, they can probably see their predator. And if they can see it, their predator is not very scary. So the ability to clear water is actually probably not super scary for them. Really turbid water is also not super scary, because in super turbid water, you're actually pretty well hidden from your predator. Maybe you can't see your predator, but your predator also can't see you. So you're pretty well hidden. It's this sort of intermediate level, the like sort of dawn dusk time, that's really scary, because you can see the shadows, you can see the movement, but you can't quite tell what it is. So that's the time when our fish were the most responsive to predators. And interestingly, it was at these intermediate levels of turbidity that our fish were also the most repeatable. And this is a pattern that we see sort of across the board. When animals judge it to be more dangerous, they're also more repeatable. So it seems like predation is really important to understanding this sort of predictability of behavior. A lot of the things I've mentioned are different measures of stress. So things like a predator is very stressful. And there is some sort of theory thinking about how just stress in general might affect repeatability. And it's actually sort of a complicated relationship, because stress can both reveal or mask variation. And as we know, variation is sort of this key measure that allows us to determine repeatability. So if we have a bunch of fish swimming around and a predator comes, we might expect that all of the individuals swim away super fast. And that would mask variation. They're all behaving the same way because they're all scared of the same predator, and there's one thing you can do: gotta swim away. However, the predator could also come in and reveal variation if individuals differ in their performance. So if some of them can swim fast and some of them can swim slow, and some of them are like, oh, there's a predator, I don't think I can outswim him, maybe I'm better trying to hide, we actually might see more variation than we would see in a non-stressed fish. So I got this idea of thinking about how variation affects how stress affects repeatability became really important to me when I was thinking about, okay, what is the developmental environment? How does that affect how they behave? And what does this really mean when the world is changing and animals are stressed out? So all of these ideas were sort of bopping around in my head. And then I was like, okay, well, maybe developmental stress is particularly important for them. And there's a lot of theory on that as well. There's the silver spoon hypothesis, which is that individuals that are not stressed during development are going to do really well later in life because they have had all of the advantages of having a perfect childhood. But more recent work and more recent theory has suggested that's actually probably not the case, and has sort of more favored the adaptive tuning model, which is the idea that high stress or low stress isn't necessarily the best. The best kind of stress is the stress that prepares you for a future environment. So if you are going to grow up in an environment where there's nothing stressful happening, it does pay to not have stress. But if you are an individual who is going to have to deal with challenging things as an adult, it's actually beneficial to experience some stress during development to prepare you for that stress as an adult. It's a learning phase where you sort of become programmed to respond to those stressors later on. What we experience during development and what our fish experience during development can become really important for both average behavior and repeatability of these behaviors. So these are all things that I'm thinking about. And the big question I wanted to ask was, how do experiences in development shape us? And specifically, how do they affect the way we respond to change? But more specifically, I wanted to look at whether or not developmental stress shaped average behavior and whether developmental stress shaped repeatability both under normal conditions and while under stress. So I was trying to think about a lot of things. I know I've already thrown a lot of information at you that are all sort of related but disparate. And I wanted to include all of those things into one project, which was maybe a little bit overly ambitious for one grad student. But I was incredibly lucky and had a very amazing team of undergraduates who worked with me. And I definitely could not have conducted any of this research without them. So they always get a shout out before I even start talking about my research, because this would not have been possible without them. So for this project, I brought mosquito fish into the lab, and mosquito fish are great because they are super environmentally tolerant. They love to breed and they love to have babies, and they are live bearers. So my female mosquito fish would do all of the hard work of taking care of the eggs, and then would just pop out little babies that we would collect right away and then divide into different treatments. So we had 4 treatments, one of which was our control, and those fish were not exposed to anything stressful during early life. Then we had our predator-exposed fish, and those predator-exposed fish were given the very scary scent of largemouth bass mixed with alarm cue, which is basically the scent of an injured mosquito fish. We had fish who exposed changes in temperature over the course of the day, so they got a huge spike in temperature that then gradually decreased. And then we got fish that were exposed to alternating predator and temperature. So they got predator exposures on one day and temperature exposures on another. And I looked at all of these different conditions because I was really interested in sort of developmental stress generally. So I knew that predators were important, but I wanted to know, is it something about predators, or is it something about just dealing with challenges that might explain patterns in later behavior? And I wanted to know also whether having a variety of different challenges was more beneficial than just being exposed to one challenge that you may become really good at dealing with. Because again, I really wanted to know whether there were sort of things we could expose our fish to that would make them generally good at responding to change and would sort of improve the way that animals are adapting to challenges. So they were exposed to these challenges for the first month of their life, and then they got 4 months where they just got to grow up unencumbered and not bothered by things. And then the first thing I did was I measured their activity, and I measured all of the fish's activity twice at one of 2 different temperatures, a standard temperature and the elevated temperature that the temperature and alternating fish were exposed to. And what we found is, first we'll just look at their average activity, and we're measuring that in terms of distance moved in these large arenas over a 15-minute trial. And what we found was that there's maybe the alternating fish are moving slightly more, but there was no real changes. They were all moving about the same amount. And this was a little bit surprising to me. Previous research has suggested that predator exposure can reduce activity in environments that fish find stressful. And I would have thought the new tank would have been a little bit stressful to them, but it seems like my fish are all behaving pretty similarly in that tank. They're moving similarly high amounts. The fish that were tested at an elevated temperature moved slightly more, but not really. But that was also a pattern that was consistent across all of my groups. So it doesn't seem like the developmental treatments really affected their average behavior that much. But what I spent a lot of time thinking about is what was happening with terms of repeatability. So I wanted to know whether these developmental treatments had affected how consistent a fish was, because I wanted to know whether these were things that we could use to understand general patterns of behavior in animals. So when we look at the repeatability, it's sort of a mess. We see things and we're like, well, there's maybe some slight trends, but there's a ton of variation around my repeatability measures, and it's really hard to know what's going on. So, there's actually a movement in the field to not look at repeatability as this overall number, because as we talked about in my introduction, the repeatability is actually a component fraction of two different things, right? There's the individual within an individual variation and the among individual variation. So you can have very similar levels of repeatability for very different reasons. So there's a movement to look at those specific measures of variation to understand patterns of what's happening. And I think that also helps make sense a little bit more of what we're seeing with the repeatability. So we're going to spend more time focusing on those actual just measures of variation. So we look at our variance components, and I'm going to spend a little bit of time on this graph because we're going to look at a ton of these as we progress through the presentation. You'll see that for each of my different groups, they're a different color, and the solid circle is the within individual variation. The striped circle is the among individual variation. And if you remember back from our TV example, we want the differences but among our individuals to be really high and the differences within an individual to be pretty low in order to have high repeatability. And what we're seeing is that in these three groups, the predator, temperature, and alternating exposed fish, we see pretty similar patterns of the relationship between our variables, that the within and the among individual variation is pretty similar. And that's giving us a slightly higher, not significantly so, but slightly higher measures of repeatability. The interesting pattern that we see is that these control fish, we're not really seeing that decrease in the within individual variation. So even though we see similar levels of among individual variations, there's similar sort of spread of different options among fish, the within individuals, they're behaving very differently from one time point to the next. And that might be important because it seems like it means that these control fish, the fish who weren't exposed to anything stressful during development, might be behaving more erratically as adults and less consistently. And if we remember, consistency is really important. It has all of these benefits for sort of group living in fish and predator avoidance and mate choice. So behaving consistently as an individual is actually pretty important. So then I also measured repeatability, again, just of their activity at an elevated temperature. So I was measuring them at 35 degrees. And what's interesting is that, again, here, there's huge variation. There's a lot of variation around my repeatability measures. I was testing between 50 and 60 fish in each of my treatments, which was pretty high. But considering the number of different things we were looking at, we probably have a lot of variation because the sample sizes realistically could have been a lot larger to really get at some of these questions. That's why it's helpful, I think, to sort of look at these patterns to understand where useful next steps might be. Because when we look at the repeatability of elevated temperatures, we see something different than the patterns of variance components at a standard temperature. We see similar measures of the among individual variation, but now even in our fish that were exposed to stress when they were younger, we don't really see a dip in the within individual variation. Now all of our individuals are just behaving a little bit less consistently and more erratically when tested at a higher temperature. And this was super interesting to me. And part of the reason it was interesting is that I'm not sure why that is. There's sort of two main reasons it could be. One is that it's beneficial to be consistent, and that at a higher temperature, they're stressed out and unable to behave consistently, that there's something that's preventing them from behaving consistently, that they're just sort of dealing with a lot of things and behaving erratically. But it could also be that when they're in a new stressful environment, that it's actually beneficial to behave less consistently. If something changes, it might be beneficial to sort of try out different behavioral strategies and figure out which one works best for you. Should you be swimming faster at a higher temperature? Or is that depleting your energy? Or is it sort of beneficial to take advantage of the fact that it's hot and you're an ectotherm and your metabolism's all sped up and you've got all this supercharged energy, and to maybe like get extra food to support that? But if there's not the availability of food, so there's all of these trade-offs that a fish might be dealing with to try to figure out what a new optimum is. And so behaving less consistently may allow them to sort of sample this behavioral space. But I can't really tease apart whether this is something that they want to do or something that they're unable to do based off of this study alone, which was incredibly frustrating and something that we will talk about at the end of my presentation. Overall, sort of what I concluded from this was that the developmental experience didn't shape their average behavior, and that was pretty conclusive, but it does seem to have some influence on repeatability, but what that influence is a little bit complicated, and there's still just like a ton of variation overall in all of my groups. And the effect would depend on the context that I was measuring their behavior, so whether or not they were being tested at a standard temperature or an elevated temperature. Then we're going to talk briefly about one more topic before we tie in the last sort of part of this experiment, and that is priming. So if you remember from the beginning of my presentation, again, I know I said a lot of things at you. The thing I was really interested in was understanding how these experiences affect the way they respond to change, and specifically how that interacts with how consistently they're behaving. So a key to this is this idea of priming. And priming is just the idea that if you are a fish and you are exposed to something like a predator, the next time you see that predator, you are more responsive to it because you've seen it in the past. You know, oh, that's dangerous. I've seen it before. But there's a lot of things we don't understand about priming. And a lot of those relate to sort of how generalizable the effect of priming is and sort of how long lasting it is. So for example, if you're a little mosquito fish and you got BASQ when you were little, does that mean that you are also responsive to another predator? So are you responsive to a trout if you've had experience with a bass? And are you generalizable sort of just like within a modality? So you like know the predators are a thing? Or if you were exposed to something like temperature, does that also prepare you to deal with something like a predator? So if you just know the world's scary, it can get hot, are you also more responsive just generally to stimuli, whether or not it's something you've had experience with? And I was interested in whether or not exposure to multiple things would help mitigate the effect of multiple stressors. So if you are a little mosquito fish who's experienced a bass before, are you better prepared at dealing with a bass when you experience it while also experiencing warm water? Because at least one of those is something you've had experience with. So that was the other reason that I was sort of using this particular design of looking at all of these different stressors on my developmental experience. So again, I've got the predator exposed fish, the temperature exposed fish, and both. And in addition to just measuring their activity, I also then measured their responses to predators. And I measured each fish twice, and I looked at their response to one of, and each fish was always tested at the same temperature, but they were exposed to both a bass and a trout. Some got trout first, some got bass first, to see whether or not, to see how these generalized their response to future predators. So these results also get quite complicated. And I will say, sort of as a spoiler, before we look at any of the data, we're going to go through it step by step because there's a lot of things going on with this graph. But one of my spoilers is that none of my treatment groups behave differently from another treatment group. You cannot tell the differences between them. There's no statistical differences. However, some of them reduce their behavior and some don't. So the thing that we're going to specifically be looking at is whether or not the fish, if the fish are behaving differently from zero, because what this is a measure of is their change in time spent moving in the 30 seconds after a predator cue is entered into an arena. So each of my fish was housed singularly in a tank. I inserted predator cue and I measured their behavior before and after that predator cue. And we're looking at just those initial 30 seconds, which are sort of the really key about whether or not they're going to be predated on, or whether the first fish is going to be predated on. And so it's a key sort of time period to understand how they're responding to these predators. And if they reduced their behavior, which from having done some predation trials with fish, reducing their behavior is the most effective way of avoiding being eaten. If they reduce their behavior, they would be different from zero. So that's the thing we're going to be looking at. It's just whether a group is different from zero, not whether they're different from other groups. So first, we're going to look at how they responded to bass. So if you remember, both my predator and my alternating fish were exposed directly to bass cue during development, and that was paired with a very scary, the smell of sort of dead fish to them. So they should be learning that that's a scary scent. Bass is also a predator that these fish have had evolutionary experience with. So there may be some sort of innate response to bass, just because this is a thing that is eating them all the time in their evolutionary history. So first, we will look at our control fish. They're slightly decreasing their behavior, but they're not really different from zero. However, my predator and my alternating exposed fish, so those fish that had experience with bass, are reducing their time spent moving in response to a predator cue, in response to that bass cue. So I was like, great, okay, they're at least, the priming worked. They are sort of remembering this bass cue and they're reducing their behavior. Interestingly, my fish that had been exposed to elevated temperature when they were little were also responding to the bass, unlike the control fish. So it does seem like the control fish not decreasing their behavior significantly, but the temperature fish were reducing their behavior in response to the bass cue. All of these is when they were tested at a standard temperature, so when they weren't dealing with multiple challenges. But then I also measured how they responded to bass cue at an elevated temperature. So when they were dealing with the stress of being in warm water. And my temperature exposed fish, so fish who had dealt with temperature change, maintained their responses to bass. So it does seem like there may be an effective priming that makes you better able at dealing with multiple stressors, so you're able to maintain your responses if you've had experiences. My predator exposed fish follow the same pattern that they've had experience with bass, and now they are also still reducing their behavior even at an elevated temperature. And my control fish are still not really responding to that bass cue at the elevated temperature. Interestingly, my alternating fish, so the fish who had experience with both bass and warm water, are now no longer reducing their behavior in response to the bass cue at an elevated temperature. And I have a theory for why that might be, but we're going to talk about it once we talk about the trout data, because it sort of relates to what's happening here. The trout cue is something that none of my fish have had any experience with, and they may have had some recent experience with trout, but they don't have a deep evolutionary history with it. As a species, they may have had recent experience. My fish as individuals have not had any experience with trout water or trout. And here we see that really that none of the fish at standard temperatures are reducing their behavior in response to trout. So they're not super responsive to the trout cue, despite reducing in response to bass. So it doesn't seem like they're generalizing, even if they've had exposure to bass, it doesn't seem like they're generalizing to other fish predators, like a novel predator. The temperature exposed fish, with the ones who've been exposed to 35 degrees, are not reducing their behavior at elevated temperatures, so that's not changing. The predator exposed fish following a pretty similar pattern, they're also not reducing their behavior. Interestingly, now my alternating fish, so fish who were exposed to both temperature change and bass cue, are really reducing their behavior in response to trout cue at an elevated temperature. And it was this sort of pattern of my alternating fish that were sort of not responding to the bass, but they now are responding to the trout at elevated temperature that got me thinking. And this could be my sort of pet theory about this that I think needs some further thought, is that when I was exposing them to alternating cues, I was always giving them on alternating days. So when they got bass cue, they never got warm water. And when they got warm water, they never got bass cue. And there's some evidence in other fish species that learning is very context specific. And fish can learn things like the time of day that a predator may be dangerous to them. And so it's conceivable that they're also learning something about temperature and when fish might be dangerous to them temperature-wise. So if my alternating fish have had a ton of experience with warm water and I know bass are a thing, I know how to deal with warm water, but I also know when the water's warm, the bass never come around. And because fish have different thermal tolerances, this is a very conceivable thing that predators may be more or less dangerous to you depending on the temperature and your relative performance at those temperatures, which is something that Alex knows a lot about. And definitely listening to her research, I think, has sort of informed some of my thinking about this. However, trout, they don't know. So trout may be that they've never had experience with this thing. They know predators are a thing, but they don't know when that trout is dangerous. So at that elevated temperature, they may be more reactive to something new because that may be a predator that is dangerous in this environment. Again, I think it needs more study, but I think it's an intriguing possibility. Of course, the thing that makes all of this even more frustrating, and that may not be at all what is happening, and that may just be that I'm getting a lot of noise in my data, is that my control fish were also pretty responsive to the trout at an elevated temperature. So those fish that weren't really responsive to bass at any temperature, weren't responsive to trout at a cooler temperature, have now become pretty responsive to them at an elevated temperature, which is very frustrating. I don't have a great answer for why that might be. But it got me thinking, as most things do. So when I was going through all of this, I kept coming back to this thing that developmental data is really hard to interpret. So when I was talking about my repeatability measures, I talked about how I can't tell whether they're becoming less repeatable at 35 degrees because they're incapable of behaving repeatable or because they're trying to sample their environment. Here, I can't really tell, are my fish reducing their behavior in response to these predator cues because they think they're dangerous? Or are there some fish that know, yeah, I know bass are dangerous, but I also know I'm really fast at higher temperatures, so I don't need to reduce my behavior. I think I can outswim the predator now. I don't have a great idea about whether they think something is dangerous or they're just incapable of responding. So let me give you an example of this. I'm running out of time. We're going to skip this section. That's just actually an even further complication that we don't need to talk about. So if we have our little fish and a predator comes by, I cannot tell if the fish sees the predator and wants to swim away really quickly, or if it sees the predator, wants to swim away, but is incapable of doing that because it's slow. So I would measure those and be like, well, one of them swam really fast, one of them swam really slowly, they're behaving differently. But those differences may be masking something about their underlying ability to do something. So this idea of what they're motivated to do versus what their capacity to do something at becomes really important when you're trying to understand how an animal is responding to something novel. And one way to tease these two things apart is that behavior is a really good idea of looking at what they are, just what they're doing. But in order to study these sort of underlying motivations, I got really interested in thinking more about physiology. And that's actually where sort of the thing that led me to do this postdoc is I moved to Scotland in order to work with Shaun Killen, specifically looking at physiology and the way that it incorporates with behavior. So some of the work that he has done looks at how metabolism relates to social behavior. So these things are really related in ways that I had no idea when I was just studying sort of what is the whole animal doing. Quick story about this. If you have a little fish who is swimming through an environment, basically their whole life is navigating this exchange between predators that want to eat them and little things that they want to eat. And this trade-off between trying to spend time not being eaten, but still spend enough time finding things to eat is a really key driver for how fish spend time in groups. And these were all things that, from a behavioral perspective, I was pretty familiar with, thinking about my fish as dealing with these trade-offs and they're making their decisions. But when you look at fish from a physiological perspective, we know that fish are all very different. So if you measure a fish's metabolism, you may have fish that have really high metabolisms that are supercharged. And they're like, I can swim fast. I'm not worried about that predator, but man, I'm hungry. I need to spend a lot of time looking for food and I don't want to compete with other fish. Versus fish that have lower metabolisms that are much more concerned about being eaten. They have lower metabolisms. They don't need as much food to fuel themselves, but they're also not quite as fast. So that predator becomes a much bigger driver for them. And being in a group gives you all kinds of protection from that predator, but does increase your competition for food. So you tend to see fish that have lower metabolisms as being more social, and fish that have higher metabolisms as being less social. And this was something that Shaun had done a variety of research on before I got here. And a lot of that was done in European minnows. And we found that, yes, this is a pattern that holds true, that we tend to see these patterns in fish. And something that I got really excited was thinking about ways that we could use physiology to better understand what a fish was capable of and how that influenced what their actual behavior was. So the big drive of my postdoc has been to better connect physiology and behavior to disentangle these questions of motivation and capacity. So the project that I have, that we wrapped up in December and are currently analyzing the data for now, is focused on understanding how acclimation temperature influences the relationship between metabolism and social behavior over changing temperatures. So ultimately, my dream is going to be to sort of use these techniques to better understand developmental stress and the way that it affects adult behaviors. But I'm now sort of optimizing my postdoc by looking at just sort of recent experience and not having to wait for my fish to grow up, but to look at recent experience and how that influences the sort of relationship between what your physiological capabilities are allowing and what you are actually doing. So for this project, we brought a variety of minnows into the lab, and we housed some of them at 20 degrees, some of them at 17 degrees, and other fish at 14 degrees. After a month and a half of acclimation to these temperatures, we then measured a variety of different traits. So we measured each fish's maximum metabolic rate. So we chased them around and then measured how much oxygen their body was using. So what is the maximum amount that their metabolic rate can get up there? And we measured that at their acclimation temperature. We measured the standard metabolic rate, which is their metabolic rate sort of in the middle of the night when they are asleep, and it's just sort of their baseline, what they need to get by. And then we measured their routine metabolic rate while we ramped the temperatures. So their standard metabolic rate was taken at the temperature they were acclimated to, and then we brought them all gradually down to 14 degrees, and then over the course of the day, gradually ramped their temperature up and measured their metabolic rate at 14, 17, 20, and then also 23 degrees. So we have really good ideas of what their metabolism is doing at all of these temperatures. And part of why we're looking at this is, right, because fish are ectotherms. They're really affected by their environment. And so fish that are in warmer water tend to have higher metabolisms. So what we wanted to know was whether or not these fish that are acclimated to warmer temperatures have a just sort of baseline higher metabolic rate, and whether that carries out across all temperatures. And then specifically, we wanted to know whether or not these acclimations to different temperatures could influence their metabolism and what effect that had on their social behavior. So is it just that when you're at a higher temperature, you all have a higher metabolism and now you're all less social? Or is there some type of like rank order conservedness such that you still have sort of the same range of social behavior within each temperature, and now the fish that have like the highest metabolism at that high chunk are the most or the least social, and the ones that have the lowest metabolism that still may be higher than other fish become sort of less social relative to their counterparts. So in order to do this, we also measured 2 measures of social behavior. We measured an individual social behavior over ramping temperatures, and we put fish into groups of 5 and measured how cohesive they were, again, also over ramping temperatures. So for each fish, we know how social they were and what they did in a group and what their metabolic rate was over 4 temperatures. Unfortunately, for the behavioral measures, we don't have 23 degrees. Our fish were not fans of the 23 degrees, and we could get metabolism rates, but the behavior data didn't work out, unfortunately. So we are actively analyzing all this data. It was 24 times 20 fish that we ran through all of these different tests, and they all have multiple measures. So we have been going through lots of behavioral videos and lots and lots of oxygen data. So unfortunately, I don't have clear conclusions yet, but I will say that looking at their individual social behavior, despite the fact that fish likely, because they're ectotherms, have different metabolic rates depending on their acclimation temperature, we see really similar social behavior across all temperatures, regardless of acclimation group. So we see variation within each group, but that variation is pretty similar from group to group. So the most social fish in each group are about as social as the most social fish in other groups. So it does seem as if it may be, again, we don't have the metabolic data all processed, but it seems like there's something happening that fish are maintaining differences in social behavior despite changes in temperature. But I'm really excited to get this data processed because I think it's going to do a much better job of understanding sort of what the insides of the fish are doing and what it's actually expressing at a behavioral level. So I would love to take some questions, but first I just want to say a couple of quick thank yous, both to my dissertation committee, Kate Laskowski, Damien Kalut, and Tajim and Andy C, and the entire C lab, and then my entire new lab here, but with a special thanks to Daphne, who is pictured here, who is a fellow postdoc that has made everything about moving to Scotland more enjoyable, but mostly the research and just life. And then Belle and Zoe, who are 2 totally phenomenal undergrads who came in on weekends and worked very long hours with us down in the basement. So thanks to all of them. And then with that, I would love to take any questions if anyone has any. Thank you so much.
Thank you so much, Amelia. I appreciate it. For everybody online, if you have any questions, feel free to put them in the chat or raise your virtual hand and I can unmute you and can ask those verbally. Amelia, you're getting all sorts of claps and thank yous in the virtual world, even though you can't hear it. So thank you very much. For everybody online, I also just want to point out the fact that Amelia is in Scotland and it's after midnight at the moment. So she is dedicated to be here with us. And we are so thankful that you took the time. So while folks are putting questions into the chat, I had a question for you that maybe is a little bit outside your research, but you talked a bit at the very beginning around individuals' response to climate change. And I'm curious whether you did any work around, I mean, it was individual and a little bit of group, but what about population?
And how populations are responding to change?
Yeah, and how that differs from, the individual versus the group versus the population, just kind of thinking along those lines.
Yeah, and so that's actually something sort of generally thinking about populations. So I sort of have 2 things to say for that. One is that like when I'm thinking about my how my variation is changing based off of whether or not you're testing an individual at an elevated temperature or not. I think this difference in whether or not it pays to have variation can change depending on what level you're looking at, right? So both in the way that an individual may benefit from behaving sort of less consistently, it's also probably good for groups generally to just have variation following change. So if you can behave less like your neighbors following change, that can be beneficial. If groups do that, those groups are the ones where some individuals are going to survive. However, the population also affects this, that you may have just different baselines for where they're coming from. And that's something that like when part of this, my final project that I was talking about, where we had acclimated our fish to different temperatures, that's one of the things that we were thinking about. So populations that come from warmer areas So populations that are starting at 20 degrees versus a population that comes from a lower temperature area have different baselines. So then when temperatures increase, are they all moving the same way? Or are you sort of at your thermal maximum at 20 degrees and you're just not going to, like, there's no place for you to go? Or the fact that you're already used to warmer temperatures, are you going to do better dealing with those changes? So thinking about these like different starting points and what that means for how individuals are changing together is something that I've spent a lot of time thinking about, but don't have a cohesive answer for what it means necessarily.
Yeah, fair enough. I was just curious to explore that a little bit. So we have a couple questions in the chat. One of them is from Alex, so you can blame her. But how are you measuring social behavior?
Yeah, so I have the chat open also, so I can look at that. So the answer to the easy question first, which is how are you measuring social behavior? So the social behavior one is we gave fish the option between a social group of 10 conspecifics or none, and just measured where they're spending their time. So that's the individual measure, just where an individual is spending his time. Does it pick fish or does it pick being alone? And then we also just put groups of 5 fish in a tank and are measuring their cohesiveness over a 15-minute period. So are they spending time really spread out, or are they spending time clumped up together? With the idea that fish that are more social are probably more clumped together. So if you have small differences in individual social behavior, if you're all slightly less social, that can actually sort of manifest in really big differences at the group level if all of your individuals are less social versus all of them are more social. So that's why we measured both things and how we were measuring them. The other question was, can you explain maybe again why you changed ramping metabolic rate and not maximum metabolic rate in the last section? Is that like why we didn't measure maximum metabolic rate at each temperature? Yes. Okay. So that's a great question. Originally, we were going to measure maximum metabolic rate at both temperatures, and it came down to a trade-off between realistically how many things we could do for our fish and what feasibly we could do and what our fish could feasibly do. So the way that we were doing it was that we would chase our fish, put them, so I can go to, this is our respirometry setup. And for people who don't measure metabolism, I had never done it before I moved here. I think this is like the coolest thing, both because it's all color coordinated, but it's also just really cool technology. So what you see here is there are little tubes, and in each tube, you open it up, you put the little fish in the tube, and this is what's called an intermittent flow respirometer. So there's an open phase, and during the open phase, water is being pumped up and through the tube, and then past each individual oxygen probes. It's measuring how much oxygen is in it, and then it gets pumped out, pushed back in, refilled with oxygen, and then pushed back in. And then there's a closed phase. And during that closed phase, no new water is pressed through. So the little fish that takes up the most amount of space that we can get in this little tube is depleting the oxygen in the water. And if their metabolism is really active, they're depleting that oxygen super quickly. So higher levels of oxygen depletion means they have a faster metabolism. And we measure for the standard metabolic rate, we measure multiple runs of these open and closed phases to get the average level of depletion over a really long time period. For the maximum metabolic rate, we are just measuring the steepest depletion of oxygen over a 2-minute period. So what is the fastest that they're able to deplete that oxygen? That's their maximum speed. So the way that we did it is we chased our fish around until they were exhausted, put them in the little thing, measure their metabolism, and then we basically tuck them in for the night. We cover up the little thing, we make sure there's air flowing, and then we turn off all the lights. Nobody's allowed to go in. And all night long, we measure their standard metabolic rate at their acclimation temperature. So they're being kept at the temperature they want to be kept at. Then first thing in the morning, we would come in gradually reduce the temperature of the respirometer down to 14 degrees. And then they would all ramp from 14 to 23 degrees over the course of the day. And we would get these respirometry measures for that whole time period. So originally, we're getting the maximum metabolic rate, the standard metabolic rate, and the ramping metabolic rate sort of all in one go, and we're never taking the fish out of the chambers. In order to measure their maximum metabolic rate at different temperatures, we would need to take them out of the chambers, chase them, and put them back in. Because for the ramping temperature, we're trying to get their sort of resting as much as we can. We don't want to stress them out before getting that temperature. So we can't measure their maximum metabolic rate during the course of the day. So originally our plan had actually been to measure that, give the fish 2 days off, and then measure them to get their maximum metabolic rate and their standard metabolic rate at each different temperature. So we would raise them up or lower them down to a different temperature, remeasure those things. So we would have even more data for each fish. But unfortunately, it turns out that when you are chasing a ton of fish, putting them in respirometry setups, and then trying to get them into behavior setups, both logistically, it was a nightmare. This was the time when we were running like 13-hour days, 7 days a week for 2 months, and like trying to also measure all of those fish's maximum metabolic rates at every temperature. There just wasn't enough hours in the day to get all of those things measured. And our fish were also, like, by the time we were running their third maximum metabolic rates, the fish were like we couldn't get them to sleep. We couldn't get them to chase. They were just like not reacting anymore. I think we just like exhausted them. So we ended up, for the sake of ourselves getting enough sleep and the sake of our fish not being exhausted, we didn't measure their maximum metabolic rates at all of the different temperatures.
Yeah, I think that sounds fair, both for you and for them.
Yeah, and I think having their maximums at different temperatures would be super interesting, but having their what their just baseline is super helpful at all of those different temperatures, because it's the metabolism that they are likely exhibiting when we're measuring social behavior, particularly because we're not as concerned about what is the max they can get up to when they're super stressed. We're not interested in predation responses. We're interested in just when they're swimming around, what is their metabolism at these different temperatures, which, considering we're just looking at what are they doing in a group when they're not stressed out, is a more useful measure for us to have.
Yeah. For everybody online, thank you so much for joining us. Emily, thank you. Any last comments before we let you go to sleep yourself after this very long day?
Okay.
All right. So again, thank you so much for joining us, Emily. I appreciate it. For everybody online, I hope to see you either tonight or next week. And we just thank you for being here. All right, everybody. Emily, go to sleep. And for everybody else, till next time, talk to you later. All right, Emily, I'm going to go ahead and end the meeting.